I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with synthesis of N-acetyl glutamic acid. In today's session, we will start with glycoproteins, in which 8 monosaccharides are commonly found. Then we have application of glycoproteins in the body, which are antigens of blood group substances and selectins, integrins, and ICMs in blood vessels. Then we have certain related disorders like leukocyte adhesion deficiency, paroxysmal nocturnal hemoglobinuria, rheumatoid arthritis, and viruses. Good morning, everyone. Let us start the topic. Glycoproteins. The glycobiology is the study of the sugars in health and disease. The glycome is the entire component of sugars in the cell. Glycomics is the comprehensive study of the carbohydrates in its entire aspects. Lectins can be used to isolate and purify glycoproteins. Glycoproteins have a protein backbone to which oligosaccharide groups are attached. Only 8 monosaccharides are commonly found in side chains. Some of them will be further acetylated or sulfated. These are glucose, galactose, mannose, N-acetyl galactosamine, N-acetyl glucosamine, xylose, fucose, and N-acetyl neuraminic acid. Depending on the sialic acid content, the electric charges are also varied. Glycoproteins are widely distributed in tissues. All plasma proteins are glycoproteins. In glycoproteins, the carbohydrate groups are attached to the peptide chains by the following types of linkages. Type A linkage through the amide group of asparagine to N-acetyl glucosamine, that is N-glycosidic linkages. The biosynthesis of N-linked glycoproteins involves dolicol phosphate. Dolicol is a polyisoprenol containing about 20 repeating isoprenoid units. Dolicol is activated to dolicol phospho N-acetyl glucosamine. This is the starting point over which other carbohydrates units are added. Tunicamycin specifically reduces the N-glycosylation by inhibiting the transferase, which adds N-acetyl glucosamine to dolicol phosphate. Here in this diagram we can see that unicamycin specifically inhibits N-glycosylation in endoplasmic reticulum. Through the linkage B, through hydroxyl group of serine, threonine, hydroxylysine, and hydroxyltaurine to N-acetyl glucosamine, galactose, or xylose. Glycosylation occurs in the Golgi bodies, and proteins that are glycosylated and properly folded are exported. Linkage 3, linked to the carboxyl terminal amino acid of the protein via phosphoryl ethanolamine, which is linked to a phosphatidyl inositol, which is in turn linked to a glucosamine in the glycan group. This is called glucosyl phosphatidyl inositol linkage. Some proteins are anchored to the plasma membrane by glycosyl phosphatidyl inositol proteins. There can be many, usually 20 to 30 oligosaccharide chains attached to a single protein. Each sugar contains may range from 1 to 20 in length. In this diagram, we can see GPI, or glycosyl phosphatidyl inositol, which is linked between plasma membrane and protein. Enzymatic addition of specific carbohydrate by a unit on the protein is called glycosylation, whereas non-enzymatic spontaneous addition is called glycation. proteins with identical amino acid sequences but having different oligosaccharide sequences are called glycoforms. The glycoproteins function as recognition sites on cell surfaces. The carbohydrates containing uppermost layer of plasma membrane is known as glycocalyx. Coming to blood group substances. The RBC membrane contains several antigenic substances based on which person can be classified into different blood groups. More than 160 different antigens are known. Of these, ABO system and Levy system are known to involve glycoproteins. ABO system is associated with three blood group substances on RBCs designated as A, B, and H antigens. The H antigen is the basic structure. It has the following structure: fucose, galactose, N-acetyl galactosamine, and protein. The RBCs carrying such H antigens are denoted as blood group O. A and B antigens differ from this in having additional sugar residues. In this diagram, we can see blood group substances in which antigen A has N-acetyl N-acetyl galactosamine linked to third residue, whereas in B antigen, the N-galactose only is linked to the third residue. The H locus codes for fucosyl transferase. In a person belonging to blood group A, N-acetyl galactosamine transferase is present, whereas in blood group B person, galactosyl transferase is seen. If both are present, then it is a AB group linkage or blood group A linkage, whereas if both links are absent, it is blood group O. In this diagram, we can see that there are AB and O systems along with their molecular structures. Coming to the selectins, integrins, and intracellular adhesion molecules. Leukocytes generally pass over endothelial surface of blood vessels. When necessity arises, leukocytes can attach over the endothelial surface and then pass through junctions between endothelial cells so as to reach extravascular compartment. Here in the diagram we can see neutrophil is transendothelial migration through endothelium where ICM is attached, and through neutrophil where selectin or integrin is attached, and finally leading to basement membrane penetration. This attachment involves interaction between selectins and integrins on the leukocytes and ICM, that is intracellular adhesion molecule, present on the endothelial cells. The selectins are glycoproteins and the corresponding receptors on the endothelial surface will have lectin type of domains. There are different types of selectins and integrins. Coming to proteoglycans. Proteoglycans are complexes of glycosaminoglycans and proteins. As the carbohydrate content is increased, viscosity is increased and solubility is decreased. The carbohydrate content of mucins is generally more than 50%. Mucins consist of 5 to 10% of mucins. These monomers are further linked together by disulfide linkages to form oligomers. The mucins will form a protective barrier to the epithelial surface. They are also found in secretions of gastrointestinal, respiratory, and urogenital tract. The glycosaminoglycans containing different repeating disaccharide units are covalently bound to the peptide chain to form proteoglycans. Coming to the biosynthesis of glycoproteins. The biosynthesis of glycosaminoglycans is taking place in endoplasmic reticulum and Golgi bodies with the help of specific glycosyl transferases. As the proteins in this is synthesized at the endoplasmic reticulum, these carbohydrate units are added one by one. Certain amino acid sequences in the protein will code for the attachment of the glycans. Specific enzymes will add the glycans sequentially. This addition needs activation of sugar. Activation in this form of corresponding nucleotide sugar. For example, UDP glucose is utilized for the addition of glucose on the protein backbone. UDP glucose is produced by the following reaction. UTP plus glucose 1-phosphate gives UDP glucose plus pyrophosphate. The enzyme is UDP glucose pyrophosphorylase. There are carrier systems or transporters to transport nucleotide sugars across Golgi membrane. Termination of the oligosaccharide chain occurs following sulfation of sugar residues. The glycoproteins have having mannose 6 phosphate as the end residue will be directed towards lysosomes. Mannose phosphate acts as a tag to deliver glycoproteins to lysosomes. Defect in the correct tagging will lead to defective targeting. Calnexin and calreticulin are proteins present in the endoplasmic reticular membrane that act as chaperones for correct folding of glycoproteins and also preventing aggregation of glycans. Only the correctly folded glycoproteins are secreted out of endoplasmic reticulum. Coming to the disorders, congenital disorders of glycosylation. These are emerging groups of autosomal recessive disorders. These are multisystem disorders mostly affecting CNS with mental retardation. At least 15 such distinct disorders in this group have been reported. Leukocyte adhesion deficiency. It is due to the mutation of Golgi fucose transporter, so selectins are not correctly glycosylated, resulting in sluggish activity of leukocyte. Coming to paroxysmal nocturnal hemoglobinuria. There is somatic mutation in PIG, that is phosphatidyl inositol gene in the hematopoietic cell. The product of this gene is necessary for GPI linkage. So GPI linked proteins are deficient on the RBCs. During sleep, there is a slight drop in pH of blood, causing mild RBC lysis and by complement factors, which leads to excretion of hemoglobin in urine. Normal person can withstand this lysis with the help of GPI linked proteins termed decay accelerating factor or DAF. Coming to rheumatoid arthritis, there is altered glycosylation of circulating immunoglobulin G. These are called aglycosyl IgG molecules. These will activate complement system, causing chronic inflammation of synovial membranes of joint cavities. In this diagram, we can see rheumatoid synovitis, where bursitis, tendinitis, synovitis, and inflammation of subchondral bone are seen. Coming to viruses, many viruses were attached to the human cells to specific glycoproteins. HIV virus causing AIDS attaches on lymphocytes by means of surface glycoprotein, that is GP120 of the virus. Influenza virus attaches to hemagglutinin H, So neuraminidase inhibitors are used as antiviral drugs against H1N1. Helicobacter pylori causing peptic ulcer binds to through two different glycoproteins present on the epithelial cells of stomach. Plasmodium falciparum, that is parasite causing malaria, attached to human cells by glycoprotein on the surface of the parasite. Here ends our session. Kindly like, share, and subscribe the channel. Thank you.